What we've got here, I think this is basically where we left off, is we gotten through these vestibular nuclei, and we were sending inputs over up the vestibulocochlear nerve, which has a couple different names, but we're sending up the cranial nerve and finally getting into the brain, where now we're running into this pair of nuclei that are receiving input from the labyrinthine organs over here, and they are then going to relay that information to other brain centers or facilitate reflexes. And I think the reflexes are really interesting. So the lateral vestibular nucleus is getting all the information from the otolith organs. Those are sort of angle of head and linear acceleration kinds of things that you're detecting. It's anything basically that's not rotational acceleration, which is sort of spinning types of accelerations. But if you start to angle your head a little bit, not so much the rotation aspect of it, but just we're now kind of sliding gradually a little off center. At this point, if you do that, then you have those little otoconia that start leaning over, which pulls the gelatinous structure, which pulls the little hair cells, which then would send input over to this lateral vestibular nucleus. That would then encounter neurons which would send information up to higher brain centers that inform you about information about your balance and what's going on. But also you have the potential to excite circuits that send signals directly down to motor neurons. So that if you're on a ship, for example, that's starting to rock over this way and you start to lean over this way, you immediately will have a reflexive motion of your limb to try to compensate for that. So you'll sort of step over with the limb and then stabilize yourself before you fall on the ground. And that is a reflexive action. Just yesterday, I almost slipped and fell on my butt on a little streak of ice. And sort of what's going on there is I start to lean back this way, I say, oh crap, and just about the time that I'm aware of this, I'm already having this reflexive action to kind of take a step back with one of my limbs to kind of catch myself and stabilize myself. And some of that could be due to otolith organs receiving the falling information, like, oh, I'm starting to fall, sending signals to the lateral vestibular nucleus, which sends signals to your limbs to cause you to stabilize yourself. We have similar kinds of reflexive actions that are associated with maintaining our head orientation, even if your body is rotating. So for example, if I start to do this, then my semicircular canals would start to pick up as my head started to rotate, and as I started to spin. This semicircular canal might have its fluid start to shift around relative to the semicircular canal, which would cause the cupola to bend a little bit, which would then cause reflexive actions that would send signals to my neck muscles to kind of compensate for that. So even as I rotate my body this way, I can maintain my head direction, in part due to little reflexes associated with the semicircular canals. I think it's less dramatic than keeping yourself from falling when you're on a boat, but the point is that we do have these reflexive actions coming in via the medial vestibular nucleus, which is only getting information from the semicircular canals. Both the lateral and medial vestibular nuclei also send information over to the cerebellum. Cerebellum is our big coordination center, so we need to have information about what our head's doing, what our balanced vestibular information is doing to do a good job of coordinating actions. They also send inputs to the ventral posterior nucleus of the thalamus. This is, anytime you're going to the thalamus, this is your gateway into the cerebral cortex. So this is receiving information and then rebroadcasting that information on up to the primary somatosensory cortex. So we do get information via the thalamus in that pathway. Vestibular information tends to go to area 3B, and it will go in there's basically a spot within the facial regions that receives this information within your area 3B of your primary somatosensory cortex. Okay. Any questions on that? Okay. Okay. So one really cool example, this is kind of the last little topic, that shows another of these sorts of reflexive actions that can take advantage of our vestibular system and use this to help us succeed in some kind of a task, is what's referred to as the vestibulo-ocular reflex. And this is a reflex by which we can use vestibular system information, particularly information about how our head is rotating through space, in order to automatically, reflexively stabilize our eyes on a particular spot. So I think I sort of preluded this before. If I look at the little camera that's in the back of the room that doesn't work, I can stare at that, and if I start to rotate my head around, my eyes will be able to stay fixed on that one spot in space. And the cool thing is, while there is some visual input, and there's some superior colliculus types of reflexive actions that can help facilitate this, it also can work in basically dark space, where if we shut out all the light and got it completely dark, like we're inside a cave in here, and I still fix my eyes on a particular spot across the room, I wouldn't be able to see anything. But if I rotated my head, my eyes could still maintain that fixation. And the reason is that we can use vestibular input to get an idea of how our head is moving, and use that then to correct the actions of our eyes and keep our eyes fixed on a particular spot. So the mechanism by which this happens is it's going to be the semicircular canals are going to be sending input. All semicircular canal input goes to the medial vestibular nucleus, which you can go back a slide. There it is. There's your medial vestibular nucleus, getting all of your semicircular canal information. So from the medial vestibular nucleus, we then send signals up to, via some stops along the way, via two motor neurons that control our eye muscles that cause our eyes to then move and respond to the rotational experiences that our head is experiencing. Come out quite right. So here's the picture that tries to show all this. And it's a little complicated. I'm never going to ask you to be able to draw out this whole thing. But I do want to walk through it to try to understand exactly how it's happening. So in this example, we're going to assume what happens if our head turns to the left. So here we're looking at the left horizontal semicircular canal. If we turn our head to the left, this semicircular canal will pick up that rotation, because what will happen is as it turns, the fluid that's within the semicircular canal will basically stay fixed even as we spin the canal around it. And so that'll basically drive that little cupola structure through this static fluid. That'll send signals up into the left vestibular nucleus, and this would be the medial vestibular nucleus because it's getting semicircular canal information. From there, we'll then elicit a number of different circuits that are ultimately going to be controlling these eye muscles. And the idea is that whenever an eye muscle contracts, or is stimulated, it'll contract and shorten, which is, in this case, if we stimulate this lateral rectus muscle here, that'll cause contraction of that muscle, which will cause the eye to pivot this way. If, on the other hand, we cause this medial rectus muscle here to contract, it'll shorten, which will pull the eye this way. Okay? So let's follow it through. Let's actually get to that medial rectus one. So here's left vestibular nucleus getting semicircular canal information that's informing you that you're turning your head to the left. So in this case, I'll follow this circuit first. This axon, or this collection of axons, would travel to this left ocular motor nucleus, which would then be stimulatory, which would then stimulate motor neurons that lead to the medial rectus and cause it to contract. So we're turning our head to the left, but as we do so, that's going to cause this eye here to pivot toward the right. Right? So as we turn the head to the left, if we want to keep our eyes fixed on a spot across the room, our eyes have to go right even as our head is turning left. Make sense? And that's going to do that for us. At the same time, we're sending signals that'll go to this abducens nucleus, which in turn is inhibitory in this case. So we're going to shut down any excitation going to the lateral rectus muscle at the same time that we are stimulating this medial rectus muscle. And that's going to cause the eyes, again, it's going to cause the eye to shift to the right because this is shortening and this one over here is not going to be shortening. It's going to be relaxing, and therefore it can be stretched. We also send signals over to the other side, over to the right eye over here. So we're going to stimulate this abducens nucleus over here. That's going to cause this lateral rectus muscle to contract. And at the same time, wait a minute. Why is that showing this? I think that's a mistake, actually. Because this, at the same time, this medial rectus muscle should be inhibited such that, again, this eye it should be stimulated in the lateral rectus muscle. And this, I really think, I never noticed this before. I don't know how I never did. But that really should be a minus symbol because we need to inhibit this muscle at the same time that we're stimulating this one. Okay? Sorry about that. Gosh. I feel like I need to get my pen out here. Hang on. That show up? Good. Cool. It helps with this, do the sound effects. Does that make sense though, as far as like what's going on here? So we're turning our head to the left. So the left semicircular canal sends input to the left medial vestibular nucleus, which then sends input to the abducens and oculomotor nuclei. Those two collectively are in charge of controlling the medial and lateral rectus muscles. We're going to stimulate this medial rectus muscle and inhibit this one, which will cause this eye to turn to the right. We're going to stimulate this lateral rectus muscle and inhibit the medial one, which will cause this eye to turn to the right. And so both of our eyes are going to pivot to the right even as our head is turning to the left. And it's all reflexive, it's automatic, and it doesn't even require visual input. And yet we can still have, even as, you know, as I'm up here like nodding my head around here, I could still more or less keep my eyes fixed on one spot in space without doing anything extraordinary, well, at least no conscious sort of effort, because I'm taking advantage of my little vestibulo-ocular reflex, and you can too. Any questions? Yes? I'm probably not going to have you like reproduce each of these things. You should know, what should you know? I think you should know that the abducens nerve is controlling the lateral rectus muscle in both cases. The oculomotor nerve is controlling the medial rectus muscle. I probably won't ask you that, but I mean, I feel like that would be fair game. Again, I'm not going to have you reproduce the whole thing. You should certainly know that if you're rotating your head to the left, that the circuit will manifest such that both eyes are going to turn to the right. So I hope that's a good enough answer. Like, you know, you don't have to recreate the whole thing, but you should know kind of the gist of what's going on. Yeah. No, except that concussions, what they're looking for when you have a concussion is some sort of disruption of brain function because you've just suffered brain damage. It's traumatic injury of brain, and therefore something like this may be more likely to, you know, something like this that has to be very tightly coordinated might not work as well. Beyond that, I got nothing. I mean, that's just a, I don't have an answer for you.
Semicircular, or the right semicircular canal, would that be sending the opposite, or like the opposite of what?
Yeah, so it would be ultimately causing the same excitation and inhibition up here, but doing so kind of like by the mirror image process. So it would, so this one, I mean, I guess a way to think about this is that when the head is rotating left, the left semicircular canal causes the ipsilateral eye to bend medially and the contralateral eye to bend laterally, right? And the right semicircular canal would basically cause the same set of inputs such that this one should turn laterally, this one should turn medially. I don't know if I really answered, like explained that well, but that is the idea. The result would be the same. These guys would not be competing against each other. They would be sending complementary information to both eyes. But each one does have inputs into both eyes. Other questions? Yep. Yep.
So she was talking about concussions and how they do concussion testing. And if this were, like, if this reflex were to be injured somehow,
Mm-hmm.
your traumatic brain injury, do you think that it's possible to regrow or regenerate the neurons that control this?
Yeah, I think a lot of times if you have a concussion and you're showing deficits in this reflex, I don't think that the neurons themselves are destroyed, but they may need time to heal. There may be inflammation present that's disrupting the pathway. I'm being pretty vague about this, but I certainly, I think this is a recoverable thing. People do recover from concussions usually, or oftentimes they do. So yeah, but I mean, I would hesitate to say that I don't think the neurons are actually being destroyed, so you wouldn't have to really regrow them, but they may need time to kind of recalibrate themselves afterwards. Anything else? This is kind of cool. All right, so at this point then, let's talk homework. So I'm going to turn you guys loose to start working on homework. Trying to see what time it is and my computer screen is blank. Okay. So I'm hoping that we can start talking in about 10 minutes or less. So try to sort of flip through them fairly quickly. I don't think they're terribly hard. Both. And you're supposed to identify which and then provide some explanations. So you may want to take a first shot at it. Just said it's the hardest. It's not that bad, though. Should we just do it as a group? Nobody wants to volunteer it. All right. Well, here's how I would. Okay. So first one. First one is just straight up linear acceleration straight ahead, right? So you are, your head is sitting, is here, and then all of a sudden you have this sort of lurch forward, and I guess, I mean, in a way you're slowing down, but it's a forward momentum that you're dealing with here. So you are slowing down in a linear way. I say you hold your head position, so you're not doing this, because that would screw all this stuff up. You are still maintaining your head position even as you're lurched forward because your father-in-law slams on the brakes. So in that case, the question is, what will happen? Well, we can look at these two structures. So these two structures, the utricle and saccule. The utricle, it says, is oriented horizontally. So that's kind of like the flat plane. Whereas the saccule is oriented vertically. So in both cases, if you sort of go along the horizontal plane here, in this plane, there's some sensors that are more going to be paying attention to left and right. And then there's some that are paying attention to forward, backward, sort of at this tip here. So these ones, if I sort of imagine your palm here is the utricle, there's some that are, these guys will be oriented to sort of left-right motions. But these ones would be more oriented towards forward, backward types of motions. So forward backwards is what we're interested in, right? So here we're sort of lurching forward. We're moving forward and being pulled forward or braking in the forward direction. So utricle should pick that up along that horizontal plane. The saccule is oriented vertically. So the way it would work is that some of its sensors are kind of oriented towards up-down motions, whereas others, it kind of loops around. Others are going to be oriented more towards left-right motions. So these are the left-righters. These are the up-downers. Not left-right. Why did I say that? I'm sorry. These would be forward-backward detectors because this is pointing out towards the front of the head, whereas these are kind of up-downers. So in that case, these ones, well, what's going to happen with these ones? Are these going to be responsive to a forward lurching motion? Or I think they are, right? So like I think you've got detectors from the saccule that are picking this up. You've got detectors from the utricle that are picking these things up. So I think both of them are going to be responsive to this. They're both responsive to that sort of forward, backward type motion. Does that make sense? The next one, I think, is the elevator one, right? So in the elevator, it says you're standing in an elevator and you hit the button and all of a sudden you kind of you surge upwards. Your body is surging upwards, which means you're kind of experiencing an acceleration down into the floor. So in that circumstance, it's going to be your up-down sensors that are going to be most responsive. The utricle is oriented in a horizontal flat plane like this. So it's not going to be responsive to up-down types of motions. Like the little hairs are sticking straight up, more or less, you could think of. So if they're sticking straight up and the otoconia right above them, if you shift the whole structure up or down, it's not really going to cause the hairs to bend in any which way, right? They're just kind of shifting upwards in their plane they're already oriented in. So they might vibrate a little bit, but they're not going to cause a substantial change. Whereas in the saccule over here, that's oriented vertically. If you have a sudden shift upward, those otoconia are kind of going to get pulled downward with the rest of your body, and that's going to cause the hair cells to deflect. So saccule should nicely pick up that information, but the utricle, which is oriented horizontally, really shouldn't. And it kind of says all that here too. Not up and down, but that's why. That's the explanation. After you lean your head forward while sitting at a desk and you're resting your forearm on your wrist. So this is how many of us spent homeroom in high school. At least that's what I did in homeroom. So I'd be sitting there, my head's resting on my forehead, so I'm oriented this way. Basically that means my head is facing straight downward. So it's not so much the motion to get there, it says after. So like after you've already gotten to this spot and you're resting peacefully on your desk, in that situation, what is going to be happening with your saccule and your utricle? So what did you guys put for this one, since we've already been talking? Saccule, utricle, both? Who says both? A couple people. Who says saccule? Who says utricle? So to me, I mean, you're basically looking, because you've changed your head's orientation, you're leaning forward in this circumstance. At this point, this is sort of, the gravity is basically functioning to pull in a, what would normally be like a forward-backward motion, right? So you're oriented downward, but the plane of interest is kind of like this, the effect of gravity, which is pulling you down this way. So if you lift your head back up and think about where the sensors are positioned, anything that's kind of along this axis should be stimulated. Right? So I would argue that both the saccule and the utricle can pick up this kind of orientation of acceleration. And if you're pointing like this, gravity's pulling your otoconia down this way, but that would kind of be like pulling them forward if you were looking straight up. It's that same direction. Does that make sense? So I would claim both. That's how I would put it. If, however, you're laying down on your side, you're now resting on a pillow with your eyes facing the wall. So in this circumstance, gravity's going to be acting and basically pulling all of your otoconia laterally. So if we pick back up and look at what can detect lateral motion. Your utricle, which is oriented horizontally, should do really good at detecting lateral motion, right? Because it's going to be able to detect what normally would be kind of left-right motion. If you lay down on the side, then that's left-right motion. That's the acceleration pulling your otoconia downward this way. The saccule, though, so again, so here's utricle. So here I am laying on my side. Gravity is pulling otoconia that way. That's going to be along this, what would normally be the horizontal plane if I had my head upright, but I'm laying sideways. Okay? So in that case, the utricle will be stimulated. The saccule, however, is oriented vertically. So if I pivot it this way, now it's kind of like, the force of gravity is pulling downward. Those otoconia are already kind of shifted. I mean, they're not being pulled, and they're not going to be causing the hair cells to bend in any which way, because this thing's oriented vertically. Is that okay? Like, I know I'm changing the head position on you, but that's part of what these things do, is these give us information about head angle. These guys will be really good at detecting forward, backward, or directly up and down as far as how but that's always relative to the orientation of your head. So as soon as you turn your head, then you have to be thinking about how is the head oriented. Yes, no, like, are we confused? What do you think? Got blank stares, which is usually not a good sign. I see a couple people nodding and giving me a thumbs up, and I got it. So I'm going to assume hopefully other people do too. Let's talk the next one. Maybe I should write these down. Should I write them down real quick? So this would be whoops. We got time. I want to call it a vector, but it's not really a vector. See if Microsoft Word knows this word. Nope. Okay. Are we okay to move on? Most people are nodding. Good. All right. So either I went way too slow in that, or everybody's good now after I wrote it. Okay. So number 2. Suppose that you sit in your chair. We often do something like this in anatomy and physiology, which is kind of fun. When you're it says describe what happens in your semicircular canals when you're looking at the hair cells. So really, we're looking at the cupola. Is it being bent or not? What should happen when your partner first starts spinning you? When you're about halfway through, you close your eyes. You might not perceive you're spinning anymore. Why would that be? And then C is after you stop yourself. So who wants to take a shot at this one? We can do this, right? I mean, I don't need to know like left, right. You don't have to be specific about like anything like that. But I guess the question is, do your hair cells get stimulated in this first one? Yes or no? I mean, they should, right? So you're sitting on a little spinny chair, and they start to spin you. That's rotational. So as you're rotating around and your head is starting to spin, that's going to cause your semicircular canals to start spinning, and the semicircular canal fluid will basically stay constant. So you're going to push the cupola through the canal 2 or 3 times, and over that period of time, you will detect that you are spinning at that moment. What should happen in this part B here that I'm describing, where maybe you don't even necessarily completely realize you're spinning at all anymore? Like, what's happening inside your semicircular canals? Did you figure this out? What do you think? Yeah, so at this point, you're spinning, and you're spinning, and you're spinning, and eventually, the endolymph is going to start spinning with your semicircular canal. So what's happening at this point is that you are spinning basically, your canal is moving still because you're still spinning, but the fluid starts moving at the same speed as the rest of the canal. So instead of at first, you're getting a pretty dramatic difference between the fluid speed and your canal speed, I'll say the cupola speed. But eventually you're going to get to a point, as you keep spinning, that your semicircular canals the fluid in there is going at the same rate as the cupola. So they're no longer going to be pushing on the cupola anymore. You're basically going to be detecting as straight up and down, and so you won't really even perceive, at least through your semicircular canal organs, you might not perceive that you're spinning anymore if it's a really nice constant rate. But then what happens when you feel dizzy? Like, why do you feel dizzy when they stop you finally and you're like trying to walk around and you're spinning around? Why is that happening? Like, what's going on in your semicircular canals that explains that? I think.
Because your canals stop rotating, but the endolymph keeps spinning.
Yeah. Yeah, so your canal stops because your head stopped spinning physically, but the fluid's still going in there. It's kind of like when you spin the egg and you're trying to find out if it's a hard-boiled egg or not, and you stop it. It'll keep spinning a little bit because the fluid's still going around and around and around and around and around. So in this case, you'll have stopped physically. Your semicircular canals won't be moving, but the fluid will be moving, and therefore the fluid will be pushing on your cupola and making you feel like you are spinning in that moment, and that causes you to feel dizzy. Cool, right? That's what's happening. And the other great thing to do is we were just talking about this vestibulo-ocular reflex. Go away. This reflex also get stimulated in this circumstance. So if you look at somebody's eyes when they're dizzy, and from this effect, their eyes are kind of like darting around a little bit. It's called nystagmus. And this is due to the fact that you are sort of overtly stimulating their vestibular reflex. You don't have to know the name of the thing, but it does screw around with their eye motions too when they're dizzy like this. All right, number 3. This one's cool too. It's very closely related. So the most common form of vertigo is benign paroxysmal positional vertigo, BPPV. So what are typical symptoms of this condition? What did you guys put down as typical conditions of this? I just need volunteers at this point. I don't want to call anybody. Anybody want to take a stab? What did you put down? Yeah, go ahead. Thank you. Pretty much, right? So you have vertigo. Like, I mean, you don't know, you're having a hard time moving around, you're nauseous, the world is spinning around you, it really sucks. What then is the mechanism that's behind this? What did you guys find for this? I want to spread this around. So like, who else? What did you find as far as mechanism goes? Did you find something? Agree, disagree? That what everybody else found? Yeah, like to the point that what I've even seen is that, you've got these otoconia crystals, which are supposed to be attached to the macula over in your otolith organs, but the whole structure is connected, so some of them can break off, and then some of them can make their way into the semicircular canals. At that point, you've got a little crystal over here. And it can move. It's just sort of flopping around in there, so as you turn your head, it will start to move, and that's going to cause little waves of motion of endolymph that can collide. Those waves of endolymph can then hit the cupola. And I guess in theory, the crystal could even hit the cupola, although usually I hear about it as more probably caused by little, basically motion waves of these crystals flowing around, pushing on the endolymph, which then deflects on the cupola, which is a super, super sensitive organ and can detect those little small disturbances in endolymph flow. So if that's the case, then all of a sudden you're getting these sort of unexpected, untrue indications or untrue stimulations of your cupola where it's being stimulated even though your head is stable by these little, motion disturbances in the endolymph caused by these little crystals getting in there. So, sucky, kind of a thing, because this is causing you to basically not be able to function. Vertigo is just incredibly debilitating. You can't walk around, you can't sit upright sometimes, you're just, you're nauseous all the time. It's really, really a severe problem. So, fortunately, there are some treatments. What did you guys find on treatments for this? There's even videos. If you go on YouTube, you can see this being done. Yeah, what'd you see? Yeah, so like surgeries, that's kind of like the last resort, right? This is debilitating. So if you're bedridden because of this and the other things aren't working, then yeah, maybe you go for surgery and you try to get the stupid crystal out of there. But there are these sort of, people have just experimentally or haphazardly even, or with some thought to what they're doing, tried to do these sort of sequences of head repositionings where they, I don't know the full sequence, but they start you here and then they rotate you a particular number angle this way and then up, and really like literally what you could sort of imagine you're doing. It's like one of those games where you're trying to like move the maze around to get the little pebble or the little metal ball out. That's what you're doing. You're like trying to move the head around, the labyrinth around. And as you do so, hopefully the little crystal will fall down out of the semicircular canal and get into an area where it's not going to cause any problems. Which, just that's amazing to me that it works, and it's that simple and mechanical, but in some cases this does seem to be an effective treatment. You just got to get it out of that canal. Other things? I think that's it. So any other questions about any of these questions or any of the topics we've covered in vestibular system? Getting no's. All right, well, we've got 3 minutes, but we're going to stop there. This exam is Wednesday. Yes, ma'am. I forgot. Hang on. So there was a question about review session. I am embarrassed to say I don't know how I did this. I just forgot. I always do one. Wait, that's next week. So, excuse me. I can do a review session tomorrow. I think, assuming I can get to campus and I don't get snowed in, I could do it at 3:30. That's like about the only time. But I could do 3:30 to 4:30. I'll find a room. So I'm sorry about that. I don't know how I blanked on that. I never not do a review session. So I will. I'll put it in here for 3:30 to 4:30. Again, as usual, please bring questions. I will find a room and I will post on Blackboard where the room is. And I'm really sorry for forgetting that. I can't believe I did.